So the materials you're going to need for this lab are 2 microfuges, a Sharpie. You're going to need your ampicillin, your pea glow, the plasmids with the pea glow in it, recovery broth, calcium chloride, E. coli, which will end up being grown in a large petri dish, Luria agar, and 4 small petri dishes. Along with that, you're also going to need multiple sterile pipettes and multiple sterile loops. Along with that, you're going to want a little boat to float your microfuges in, the tips for your micropipetter and a micropipetter, and then you're also going to need an ice bath and a heat bath. So the first thing you guys are going to want to do is take your centrifuges, which you have labeled positive and negative for the DNA, and then you're going to want to open those up, and at the same time open your pipette. Then you're going to get your calcium chloride, and you're going to get 0.25 milliliters, which is the same as 250 micromilliliters. Put that in one of them, and then you're going to want to open those up, and at the same time open your pipette. Then you're going to get your calcium chloride, and you're going to get 0.25 milliliters, which is the same as 250 micromilliliters. Put that in one of them. And then you're going to do the same thing in this one. And then you're going to close this up and this gets thrown away. And the next thing you're going to want to do is take your bacteria and open up one of your loops. And then you're going to grab 3 or 4 colonies, which is just the little dots that you see. And you twirl them around in there. And then this gets thrown away, and this gets closed up. And you do the same thing with your other centrifuge. So this is our ice bath, and this is our heat bath. When you actually do the lab, it'll be a little bit larger, just so you can fit more in at one time. And Mr. Edwards will be monitoring the heat of the heat bath, just make sure it stays the same temperature. So to start, you're going to put your little centrifuges into your ice bath, and they'll stay in there for 15 minutes. So, and I'm going to put it into the heat bath right away for exactly 90 seconds. Take your recovery broth, and you're going to take a sterile micropipetter, and you're going to put 0.25 in each of these. And then you might want to just put them back in here. You're going to put them back in here, and then these will go into the incubator for as long as you can put them in there for.